All right, so hopefully everyone's back by now. I had a little time to get up and stretch their legs. I'll be chairing the last session of the day today before we get to our evening keynote. But we have two more great talks coming up. And the first is by Andy Nieuwkoop, who is currently a professor at Rutgers University. Andy did his PhD work at the University of Illinois at Urbana-Champaign, working with Chad Dreenstra, doing some cool solid-state NMR on amyloid fibrils. And then after that, moved on to a postdoc position at the SMP in Berlin, working with Hartmut Ashkenat, again, doing some very cool and cutting edge solid-state NMR of membrane proteins. And after that, is now an assistant professor in the Department of Chemistry and Chemical Biology at Rutgers. And he's been working on looking at some site-specific interaction dynamics of proteins, membrane proteins, and the membrane specifically, and also pushing the cutting edge of using proton detection with solid-state NMR. So, Andy, take it away.
Thank you, John. We've already confirmed everyone can see and hear me. So, yeah, I will be talking to you, assuming I can figure out how to advance my slides in Teams, about the work I'm doing in my lab here at Rutgers. I figured out, given the length of this talk, I was going to have to cover everything that we do, not just the PIP binding project. So this is sort of the main project in the lab, where we look to understand PIP, phosphatidylinositol phosphate binding, and the bilayers that contain them as well. One of the things that kind of came up, I think, a lot in Anne's talk this morning was how important thinking about the bilayer is in addition to thinking about the proteins that interact with it. So that was definitely one of the take-homes I got from my time in Berlin that I've definitely taken forward with me as I started my own lab. And as John mentioned, we spent a lot of time working on fast magic angle spinning. We heard a lot about how you can spin faster nowadays with smaller and smaller rotors. So I've been taking advantage of that new hardware in a lot of my projects. So I'll spend a tiny bit of time talking about that. And then the last part of the talk today, I'll talk about my materials type projects. As you heard from my CV, I've spent a lot of time in my life as a bio solid state NMR spectroscopist, so a squishy state NMR spectroscopist. But having arrived here at Rutgers and I started my lab, there's a lot of really cool materials, inorganic chemistry type things happening on campus. So as I've sort of reached out to colleagues, and to be honest, as my students have reached out to their fellow students, we've actually developed a couple of pretty cool projects working on much more traditional, quote unquote, solid state NMR. And so I'll share with you some results at the end that we've gotten on those projects as well. It doesn't work. All right, so yeah, I do solid state NMR, and as my career track kind of goes down this slide here, solid state NMR is great for studying nanocrystals, so something like GB1 or ubiquitin, firals and aggregates. So we heard about synuclein in the solution this morning, and as John pointed out, I spent my PhD with Chad studying it as an aggregate, and then Anne, I guess, talked about some aggregates again later today. And then membrane proteins in the lipid environment. And there was this last part that I found really exciting, mostly because I feel like it's the place that that kind of standard structural biology falls down the most. It's the hardest to study membrane proteins in their native lipid environments, and a lot of the more static techniques, so X-ray crystal or a cryo-EM structure, might have a harder time understanding exactly what happens on a lipid membrane. So this is where I decided to focus my efforts. And then, as we pointed out, we like to spin at the magic angle as quickly as possible. And so I usually spend a little time on this, but Matthias spent a whole hour this morning describing in great detail all of the wonderful things that happen when you spin very quickly. So I will just point out that now that we can get up to 100 kilohertz, our proton alignments are getting really quite narrow compared to what was considered really great when I started in graduate school. All right, so the main research focus in the group is these phosphatidylinositol phosphates, which I'll call PIPs from now on. They make up about 10% of the phospholipids in the human cell. Maybe that's a high number. There's 7 different species of them, and they're distinguished by their phosphorylation pattern. So basically, you have this head group with this inositol sugar, and it can be phosphorylated at the 3, 4, or 5 positions reversibly. It's a very complicated and exciting regulatory network. And basically what your body does is it uses these lipids as signposts to sort of say what part of a lipid membrane you're on. So one of the things I guess we've started to realize is that a lot of biology takes place on lipid membranes. It's kind of a good place to try to do the chemistry of life. And so one of the things you need to do then in a complicated cell is identify where you want to be doing your chemistry. So you want to get things together. A good place to do that is a lipid membrane. And if you want to be controlling in a sense, biology life sense, what's going on where, an easy way to do that is through different lipid head groups. And so what happens is these PIPs are made from inositol, from PI, and then they're kind of decorated by a very tightly and complicated set of enzymes, set of kinases and phosphatases, mutations in which are known to cause large numbers of cancers just because of how important these signaling pathways are. But basically what I've been focusing on in my group is understanding how these PIPs present themselves on lipid membranes and how the proteins that interact with them are controlled by those interactions. So the idea here is that we don't understand well why a certain protein would bind a certain PIP. And so the first thing that you would think of when you see something like this is to say, well, a protein binds PIP3, doesn't bind PIP2. Well, it's a lock and key type mechanism, right? Well, there's this extra phosphate group and it'll stick in the binding site and the protein will recognize that. And one of the things that's become very clear as we've started studying these is that it's very much not the whole story. There's a lot more going on. And so one of the things that sort of made me want to go into this is that PIPs in general have a tendency not to be studied in native environments. And so I think the very next slide will even show you the fact that the easiest way to study a membrane protein that's only a membrane protein because it binds a lipid head group is to just cut off this lipid head group and study this in solution. And so basically what you do is you study inositol phosphate binding instead of the PIP lipid binding. So the problem with that, of course, is you're ignoring the context of the rest of the lipid bilayer. And so basically what we want to know is what's happening with the rest of the lipid membrane. So basically everything in addition to the small percentage of the membrane that's PIPs is also interacting with the protein. And that's going to be lost if you study it with something like SPR or solution NMR. And again, I will show you some solution NMR. So actually a great way of studying these, and we need to combine solution and solid state NMR to try to get the best out of both worlds, out of both of them. All right, so the first thing we have to do in order to do solid state NMR of these liposomes or lipid bilayers in general, since we're now studying nanodiscs as well, is to get them into the solid state NMR rotors. So one of the things we spent a lot of time on the first few years in my lab is optimizing this protocol. And so most of the lipid stuff I'll show you today is done in 1.6 millimeter MAS rotors. That's because our Phoenix HXY NMR probe, which allows us to tune the phosphorus and nitrogen or phosphorus and carbon, is a 1.6 millimeter probe. And so that's where we've been doing most of these initial lipid studies. And so the idea here is that we need to somehow get a liposome pelleted in an ultracentrifuge tube. And so basically we create our liposomes by sonification, or we create our nanodiscs off a gel filtration column, and then you have these soluble lipid bilayers, but we want to study them as compact as possible. And so what we do is we ultracentrifuge them. So usually for a couple of hours, we've played with that time a little bit to try to get the optimal packing without too much squishing. There's a little concern that if we ultracentrifuge liposomes in particular too long, we might be messing around with the bilayerness of our bilayer. But now you have a nice looking liposome pellet, like you can see here, but you can't get that into a rotor by scraping. It looks like you could try the tried and true solid-state NMR, scrape that with a, spatula and stick it in the top of the rotor, but it doesn't work. It's just really frustrating experience if you try to do it. And so what we do is we use something that Chad developed for exactly this purpose a few years back, where basically you stick the ultracentrifuge tube in the top of this container, you stick your NMR rotor in the bottom, you flip the whole thing upside down, you put it in a Falcon tube, and you spin it on a spinning bucket for a few minutes to kind of push the liposome pellet down the side of the ultracentrifuge tube and directly into your rotor. And that works really well. It gets mostly lipid with enough water that it doesn't completely dry out, and you get weird looking spectrum. So that was been the kind of the first thing that needed to be optimal before we were able to study our samples with NMR. And so we've done the same thing most recently with the 0.7 millimeter tools. So those of you who've interacted with this know that Bruker put together a really pleasant set of tools for interacting with the 0.7s. So as the rotors got smaller and smaller, the home-built remedies for getting samples into rotors, pipette tips flame sealed on top of other pipette tips and stuck into Falcon tubes. It's just how I packed a lot of rotors when I was younger. They've gotten harder and harder to interact with. And so what Bruker's done is a set of kind of hands-free tools where basically you don't ever have to grab the rotor by yourself. You're actually using tools the whole way. And so what Tom Osborne Pop, who's a postdoc in my lab, did was actually 3D print a set of tools that go on top of these Bruker tools to basically do the same thing we did in the previous slide here, just with machined materials. So these were done in the machine shop. These were actually 3D printed with their fancy new 3D printer he got as a result of his Beckman postdoctoral fellowship, actually. And so then the same story happens. You have your lipid sample in the ultracentrifuge tube, you spin it down, and it goes into your 0.7 millimeter rotor now, which you can then pick up with the normal Bruker tools and stick your caps back on. And so that's kind of been the necessary steps we needed before we could start doing the NMR we wanted on our samples. All right, so why do we want to do proton and phosphorus NMR of lipids? And the answer is, of course, for this audience, kind of clear, which is that every NMR active atom in a system gives you a signal, and that the chemical shifts are very sensitive to the environment. And so proton with a gyromagnetic ratio of 1, which is kind of awkward because, of course, gyromagnetic ratio is dividing by the gyromagnetic ratio of protons, so it's by definition 1. But basically, the highest stable isotope in terms of sensitivity, and then phosphorus also actually quite high, so higher than carbon, and 100% abundant. And so basically what I was thinking about when I was designing this sort of line of inquiry is what can I do, what tools can I use to look at interactions? And so the two that are already there without having to isotopically label anything are proton and phosphorus. And so you get these really nice resolved proton spectra above the lipid phase transition, which allow you to kind of identify all of the different regions of the lipid tail. And then the phosphorus head groups are the same. So basically what you're seeing here is the glycerol phosphate of the phosphatidylcholine that the sample's mostly made up of. So this sample's 95% phosphatidylcholine. And then the other 5% are a PIP. And so the nice thing here is that PIP3 here with 3 different phosphates on the head group has different chemical shifts for those additional phosphates than the PIP2, 4, 5 here. And so basically the idea here is that since they have different environments, they have different chemical shifts, and because of that, we'll be able to identify the reason PIP3 activates a protein, whereas PIP2 doesn't. And so that's something that, like a biologist, very comfortable with, but as a structural biologist, as a physical chemist felt very strange to me, in the sense that this is a very similar chemical molecule, right? There are not a lot of differences, and you could imagine that any interaction this molecule makes, this molecule could make as well, but that this difference here doesn't strike me necessarily as the large enough to change all the different behaviors that we see when you see these phosphorylations. So first thing we had to do was assign the protons. And so we did that kind of straightforwardly with a 2D HSQC-HC sort of experiment. I'm showing this because it was sort of amazing to me in the sense that the solution NMR, which is the blue, and the solid state NMR there, that's the red, very similar chemical shifts like we talked about this morning, but also very similar overall resolution, I guess I would say. And so one of the things that's kind of fun to think about when comparing solid and solution NMR experiments, which I've done a lot of in my life, is that while the proton line width of the solids is not as good as the solution, and so this is actually a carbon detected experiment in the solid state, so we've actually presenting it this way, as opposed to maybe the more traditional proton on the x-axis way, which is definitely the way the solution NMR would be presented if I was showing it alone. But you can see the proton line widths in the solution liposomes here are really just quite great, right? Whereas the solid state are a little broader. But the interesting thing is that the carbon line widths are sort of exactly the reverse. Because of the way you can digitize an HSQC spectrum, you actually get nicer, maybe, or have the possibility for narrower carbon line widths in our solid state experiments. And so overall, resolution is not going to be the limiting factor when we're looking at these samples in either solution or the solid state. Obviously, sensitivity becomes a bigger challenge in the solid state experiments just because we're limited by the amount of material we can fit in our rotors. All right, so what are the tools we're going to use to study these lipids? Well, the first one we like to think about a lot because it sort of reports broadly on the structure of the lipid bilayer is the lipid phase transition. It's just very clear here in an extreme sense, where if you're above the liquid to gel phase transition, the protons on the side chains are very mobile. You get really sharp signals. When you drop below that, you have much broader lines, and you can quantitate that with proton T1. So you get the T2 relaxation times. I'm sorry, I said T1, I meant T2. Or basically, you can see that below that phase transition, the proton T2s get very short. And so that's a similar situation to what was we were seeing this morning when the things were entering the gel phase, just much more dramatic. So instead of going from solution T2s to slightly less solution T2s, we're going from solid state, nice solid state T2s to bad solid state T2s. So these get really low, and of course the line widths get really broad. All right, so as we're getting feedback there, as we seek to have our best representation of the inner cellular membrane, which is actually the sort of position we've decided to place ourselves, one of the things that becomes clear is we need to include some amount of anionic lipids. So as Anne pointed out when she was talking about her K2 or her ion channel, lipid membrane composition, you need some percentage of these negatively charged lipids. And so because we're looking at human proteins, we're using phosphatidylserine. It's got a negative charge on that head group. And so that actually helps increase the negative charge of the membrane. So basically the pips we were talking about have a net negative charge of 4 or 5 or 6, depending on the kind of protonation state. Well, 3, 4, 5s may be more reasonable. But phosphatidylserine is also negatively charged. And so one of the things that we're going to look at when we look at our binding domains a lot are that there's a lot of charge-charge interactions occurring between the two. And so in order to sort of completely recapitulate the appropriate environment, we need to have something like PS in our membrane as well. And so the samples we've been making have been kind of 30% phosphatidylserine mostly, which is kind of a normal-ish number for the inner cellular leaflet of a human cell. And so the idea here now is that in addition to the phosphatidylcholine sequence backbone phosphorus, you also have the phosphatidylserine. You can see you get good resolution between those two, which is actually nice if we're going to try to think about site-specifically talking about whether something's interacting specifically with PS or PC. And of course, when you have a negatively charged, as opposed to a Zwitterid or ionic lipid head group, when you add in a divalent cation, you actually see clustering. And so this is something that Chad demonstrated in solid state NMRs, maybe the better way, since people have known about that for quite a while. While I was there at Illinois, and so basically the idea here is that when you add phosphatidylserine, you actually get nice sharp phosphorates signals, but when you add calcium, you actually start to see clustering of those phosphatidylserines, which causes a broader phase transition because the single sort of phase transition from disordered to ordered is spread out because of the increased order of those calcium sort of clusters. We don't see any evidence above the phase transition with no calcium of a PC to PS interaction. This is a DAR spectrum between two phosphorus. I'm not 100% sure I would expect a cross peak given how far apart they are, but I definitely don't see one. And then at calcium, you could maybe talk yourself into, well, a broadening of the PC line with, as you can see from the 1D, if nothing else. And there's maybe some hints of an interaction there as well. All right, so now that we've thought about our negatively charged PS, we'll start talking a little bit more about what we do with PIP3. So the protein we study, which is kindlin 2, which I'll introduce here in a couple of slides, primary binder, the lipid that activates it the most is PIP3, so that's kind of the lipid I'll focus on for most of the talk today. And we've been studying 5% of that in the membrane, which is like sort of the upper limit of what you could call physiologically plausible. I think biologically relevant might be overselling it slightly. It's really hard to say though, because these PIPs are very small percentages overall, but they can be very highly concentrated locally. And one of the things I'll show in the later slides is the presence of our PIP binding proteins actually has the potential to increase the local concentrations of PIPs. And so it's a situation where there isn't a lot of it in the overall cell membrane, but because it's such an important lipid that activates both function, but also protein-protein interactions, the idea that local concentrations of PIP could get pretty high is actually not completely unreasonable. So we have our PIP samples. You can see here that when you have just PIP3 and PC, you get really nice signals. This is the one I showed you earlier. When you add calcium to that, the signals are still here, but they're broadened out dramatically. You can expand this and vertically sense and still see clear definition between the three phosphates, but I kept the figure like this to kind of be honest about how much broadening we saw when we added calcium the first time. And then interestingly, when you have PIP3 and PS together, we actually see this fourth peak. And so there's an interaction of some flavor happening between the PS and the PIPs. Either it's created a separate environment, which is resulting in a different shift, or it's making one of a bridge or some kind. And then when you add the divalent cations, same kind of thing happens. You get much broader lines in the phosphorus spectra. And so as a result of this, most of the initial data I'm going to show with the protein binding is done without calcium, although I will show you with the simulations later on that there is a reason to believe that we want to swing back around and look at those calcium spectrum sooner rather than later. So you can see similar effects with the phase transitions, where basically adding calcium broadens that phase transition and decreases the temperature. And the inclusion of phosphatidylserine sort of sharpened that phase transition without the presence of calcium, and then dramatically broadened it with the presence of calcium. And so these sort of really broad phase transitions are sort of a hallmark of what we're seeing right now with our PIP samples. And so one of the things we're going to need to figure out moving forward is exactly what that means in the context of thinking about our interactions with our protein. So basically, if we want to understand what the surface of the membrane looks like in context of presenting these pips as binding partners, understanding what these broadened phase transitions mean in terms of the dynamics of the head group as opposed to the dynamics of the whole lipid as a whole is going to be one of our goals here for the next little while. All right, so as I've teased a couple of times, the primary protein we study interacting with pips in the group is called kindlin-2. It's a human protein, and it's one out of hundreds of proteins in a human that actually bind pips. And we've decided to focus on it because it has two separate pip binding domains, which is actually kind of interesting. So it's got a plextin homology domain, which is sort of a common, I've flipped this figure. It's got a plextin homology pH domain, which is sort of a common lipid binding motif. And then it's also got a F0 domain, which is a modified ubiquitin. It's a ubiquitin fold that also binds pips. So actually, this protein has two different places where it can interact with pips. And so that actually gives us a lot of interesting questions we can answer with regard to what we're expecting to happen when we are interacting with pip-containing lipid membrane. And so the big open questions we have about kindlin. So kindlin-2 is involved in cell adhesion. It's part of the integrin signaling, inside-out signaling pathway, where basically integrin tells you what the outer cellular structure forces might look like, and then transmits that into the cell via the actin scaffold inside. And so kindlin is one of those proteins that shows up as a little ball in those diagrams. And one of the cool things about those diagrams is they almost never show kindlin on the membrane, because you're trying to build one of those word art pictures, and it's very clear that the fact that kindlin is a membrane-bound protein is not high on most people's list when they think about this. And so the class of protein of kindlin-2, kindlin's the second of three human kindlins, and they're all firm proteins, which is where it gets the name for these domains. There's an F1, an F2, and an F3 domain, which are kind of conserved across the firm proteins. That ubiquitin pip binding is an addition, which is why it's labeled F0, and then the pH domain is just kind of plastered, modified in between two loops in the F2 domain. So there's actually an insertion in the gene, which is that second pip binding domain. And so the big open question in the kindlin field is that there was a crystal structure, and I guess I have the citation a little later in the talk, that was solved of this protein, and it was a dimer. But they solved that crystal structure by cutting off the pH domain. So you can see here we've put it back in using Photoshop. And I'll show you a slightly more accurate reassembly later in the talk. But basically, in order to get this to crystallize, they had to get rid of this pH domain. But when they did that, it crystallized as a dimer. So there's a lot of questions about whether that dimer is physiologically relevant, and there's some evidence to indicate that it is. It seems to show up in biological systems. It's some indication that kindlin-3 is always a dimer, or at least its active state is a dimer. And so there's a reason to expect this dimer is real. But of course, it was done not on a membrane and without the membrane binding domains. So there's a lot of good questions there about what we expect the effects of these lipid binding domains and the lipid binding itself to be on both the structure of that protein and then how it dimerizes. So if you have a pip binding domain that clusters pips around it, like I'll show you here in a little bit, it could cluster another pH domain together here, and you could have dimerization occurring on the membrane as a result of pip binding. So that's kind of the question we are setting out to ask. And then the other big question we're thinking about is that F0 domain here is a hub for a lot of interactions. So not surprisingly, ubiquitin likes to bind to other proteins. And so this helix here is a site of multiple protein-protein interactions. And so one of the questions we're looking into is how pip binding affects the ability of this protein to bind protein partners. All right, so like I said, the first thing you do when you get one of these pip binding domains is you study them in solution with an inositol phosphate. And so this was done for the Kindle 2 pH domain a long time ago, maybe 10 years ago. And they solved this solution NMR structure. And so this is the published solution NMR structure of Kindle 2 pH bound to inositol phosphate. And then here's our own solution NMR data, where we basically recapitulate their results. We've completed, we've confirmed their assignments, maybe made a correction or two as we went, and have able to show that the inositol phosphate binding induces changes at the expected canonical pip binding site. And so we then wanted to move on to see what that looks like in real pips instead of in solution. And so what we've done here is taken phosphorus spectra, where we showed the peaks of HUD. So I've shown you these two figures before, where basically you have PIP3 and PC, and PIP3 PC with PS, with that extra peak. And what happened the first time in liposomes when we added the Kindle 2 pH domain, we got this spectrum, which basically showed a kind of I don't know, blurring out of the PIP head group peaks, and we weren't able to get a particularly clear understanding of what phosphate that is, we had a lot of problems with Kindle 2 pulling our pips out of the membrane. So if you guys remember, or were at my talk at ENC a couple years ago, I showed some PIP2 data where the pH domain sort of ripped our pip right out of the membrane, which is apparently something you can simulate. It happens. Well, these highly charged lipid head groups are actually reasonably close to being soluble in water, so it doesn't take too much, apparently, just pull them all the way out of the bilayer. And so one of the things we wanted to do to sort of combat that / open up our options was nanodiscs. So that's the last panel here. This is data that we're recently acquiring. The sample's actually still in the magnet, where basically we created a nanodisc a la Steve Slagar with membrane scaffold protein with the same lipid compositions, although I guess in this context we don't have PS in the current sample, and actually got substantially more stable pH domain lipid interactions. So we're very excited about that. The interesting thing here is that these three spectra are all direct polarization. This is actually cross polarization because we couldn't see the direct polarization, and we're not 100% sure why that is yet. This is also dramatically less sensitive. But it's interesting in the sense that when you actually go down to the cross polarization, you're seeing primarily the sites from the PIP head group as opposed to the backbone phosphatidylcholine glycerol phosphate peak. So that's really quite interesting. We're excited about that result. We're not sure what it means yet, but we're pushing forward to try to figure out what's happening there. And in the same story, in terms of what we were seeing with the phase transition, we have a dramatically broadened phase transition. So everything I've been showing you so far has been running 20 to -20. In order to get this to even come down to 25% of the original T2, we had to go to -40, which is about as cold as we were comfortable getting our the chiller on our 600. So, I mean, this has sort of bottomed out for the last 10 degrees, but we haven't actually gone colder yet. So I can't tell you if this has a second sort of drop off where it goes down to 0 or not. So a lot of interesting results there, and I'm hopefully in the future we'll be able to tell you a little bit more about what they all mean. All right, so that was kind of the PIP bilayers portion of the talk. I want to move on now and talk a little bit about the proteins. We have some, like I showed you, NMR data there, and then there's also a lot of computational work as a result of the close proximity of Dave Case and the global pandemic that didn't allow us in the wet lab for quite a while. So as we pointed out, I've spent a lot of my career recently working on proton-detected solid-state NMR because proton has the highest gyromagnetic ratio, and you get this extra sensitivity by spinning quickly. You have to deal with those strong couplings either by diluting the protons or spinning faster, and so what I'll talk about today are the 100 kilohertz results, where you basically can get away with fully protonated protein and still get nice-looking spectra, although moving forward, I think we will be deuterating our samples to do experiments like Lewis talked about this morning, where basically if you deuterate your protein and you leave your natural abundance protons on your lipids, you can actually look at intermolecular interactions more easily than you can with a protonated protein. All right, so this is just to show off that we get high sensitivity with small amounts of sample, and basically that the overall line width of your sample does not depend on the size of the protein anymore. It now depends on the homogeneity of your preparation and how fast you're spinning. So basically, with good sample preparations, we can get down to 10th part per million proton line widths, spinning at 40 or 60 or 100 kilohertz on these samples. And it's a lot of magnet effects, I guess, as we spent a lot of time talking about. There's a lot of inhomogeneous effects that are starting to dominate as we get to higher and higher spinning rates. So here's my 100 kilohertz of GB1. I think this is the last GB1 spectrum I show, so I apologize. It's just nice to show off the prettiest data, where I basically show that the protonated GB1 at 100 kilohertz, which is the blue there, has similar line widths to the 40 kilohertz GB1 that is deuterated. So there's a little bit of cheating here because the 0.7 is an 800 and the 1.6 is a 600. So we got a little bit of magnet here, but it basically lets you use the normal quote-unquote solution assignment type experiments through the amine backbone and the backbone carbons to assign your chemical shifts without needing to use, using protons instead of using carbons and nitrogen detection, which if you already have, as I pointed out, the solution NMR assignments of your protein is actually quite helpful. And so I showed you that lipid sample at the beginning where basically we had the lipid, the solution NMR on top of the solid state NMR. And you get these little X's where the carbon resolution is narrower in the solid and the proton resolution is narrower in the solution. It's actually really helpful. You're not de novo assigning anymore. You're transferring the assignments. So this is what that looks like. So the blue spectrum I showed you earlier is pH domain in solution. And then the solid state sample is 100 kilohertz, 105, I guess, on the 800 here at Rutgers. And basically, you can see that the resolution of the sample isn't great, and we're still working on our solid state preparations. We actually have getting back on the 800 here in not very long, and we're hopeful that the nanodisc bound sample will actually give nicer looking spectra. But the nice thing was that we were able to confirm the assignments of the solid state sample using the solution NMR assignments in a way that is no way, I mean, you have strips in the backbone walk that look like this, which you could absolutely convince somebody that, yes, I see these two peaks are in the same slice and they definitely connect to each other. But you also have strips that look like this. And this isn't a strip where anybody who is, has a straight face would tell you that they could pick out which peak matches up to which peak. So like this example, the peak here in red is a shoulder on a much larger peak, right? There's not enough resolution in that solid state to unambiguously do that backbone walk, but we don't have to because we have the solution NMR backbone walk that we can overlay on top. The peaks sit right there together, and it's just really straightforward to transfer assignments using the 3D assignment suites. So you could not argue, you could argue you could transfer this assignment, for example, using just the 2D, but you need the 3D to transfer all the assignments in the middle here, where there isn't enough resolution. And so that's really helpful, and it's going to be the goal for the project moving forward, basically, where you can assign things relatively straightforward. These domains aren't that big, like the 100 amino acid, size range. In the solution, and then use those solution assignments for the solid state assignments, saving a lot of time and effort of trying to, spend 10 years getting your first set of solid state assignments, which has been like the hallmark of the really exciting biology that has been done in the solid state NMR up to this point. All right, so like I said, we're going to take those assignments and try to figure out what's happening when we bind our pH domain to liposomes or nanodiscs. And so here's that binding site I showed you from the solution NMR. As you can see, not surprisingly, the negatively charged phosphates are interacting with a bunch of positively charged nitrogen side chains. And so that's quite interesting in the sense that you can see that in a nitrogen 1D. And so this is where we kind of wander into data that is brand new. So this nanodisc spectrum is again from this weekend. And one of the interesting things that we found when we were starting on that set of experiments is that because of that phase transition diagram I showed you earlier, we are studying the nanodisc bound sample much, much colder than we ever did the liposome bound sample. And the CP efficiencies went up dramatically. We got good CP when we got really, really cold. And so this isn't shocking, I guess, as Anne sort of pointed out when she talked about KCSA earlier. Integral membrane proteins, you see this behavior where above the phase transition, you get really nice sharp lines because everything's moving. Below the phase transition, you get great CP, but you get really broad lines because the tails have all frozen out in strange conformations. And it does unpleasant things to your transmembrane protein line widths because you just end up with a lot of different chemical environments. Because the pH domain is not in the membrane, it's just sitting on the surface, we actually got much, much better spectra way below the lipid phase transition. And so that is interesting and something we're thinking about moving forward. So the big difference between this spectrum, which is all a big preface for the fact, the big difference between this spectrum is that it's at -40 and the spectrum is at 20. And as you can see, the side chain peaks are much more strong in this liposome bound sample than they are in the nanodisc bound sample. But I think that is a slight misnomer in the sense that the backbone amide CP got dramatically better on this sample below the phase transition. So I don't think these peaks are shorter. I think this peak's taller. But you can also see there's much broader distribution here on the frozen out sample. But there's actually some chemical interesting things here. I think the histidines are spread way out. But they show up pretty okay on an NH2D, which I did not show because it was taken at 20 kilohertz, and I'm waiting until we get the 100 kilohertz data before I whip that out into public. I don't want to show broad proton line widths. But basically, we're really excited about our ability to look at these side chains using the NH2Ds moving forward. So here's the carbon spectrum of that same sample, that PIP3 bound pH domain. And so something interesting happened here. First thing we did on the sample is took an SPC5 one-bound correlation spectrum. You can see that in red. We got really nice looking alanines and pretty okay looking threonines, and anything that didn't have a methyl, kind of not so great. And so after that, we signal averaged a DAR. And one of the interesting things that showed up is that residues that are directly in contact with the membrane, or hypothetically directly in contact with the membrane here in red, don't show up well at all in the DAR spectrum, where they show up really nicely in the SPC5. And then other residues kind of out in loops did the same thing. So that might be just a side effect of there being well, the protein being very cold at -40, but it was an interesting result that we're going to kind of look into moving forward, that the residues on the membrane responded better to, direct SPC5 or sort of recoupling as opposed to sort of broad-based proton-driven DAR mixing. So maybe that has something to do with there just being too dense of a proton matrix on the membrane surface, so like our signal just kind of disappeared. Think of it, maybe we just try a very, very short DAR mixing time and see what we get there. But I thought it was an interesting result, and like I said, that spectrum is from last week, so I haven't had a huge amount of time to think about it. But a cool result and a demonstration of how this is a first carbon-carbon 2D we've taken on the sample. There's no way we could get the assignments on this sample de novo, but because we have the solution assignments and we confirmed them using the 100 kilohertz data, we're able to put labels on these peaks with a fair bit of confidence. Actually lets us interpret these data without having to have taken the NCACX, assignment suite that would take weeks and weeks and weeks. All right, so now we're going to move on here to talking about some of our simulations. And so one of the things we wanted to understand is what the interaction with the membrane looks like in a sort of time average sense of the word. So obviously the solution NMR structure, kind of a static structure showing the head group bound in the primary binding site. But what we want to understand is what that interaction looks like. And so Robert Polymer in my group has been the person working on this with Dave Case. Dave Case very generously donating large amounts of his computer time prior to our ability to purchase our own. And so what we're seeing here these were working a second ago. Is a much less than static situation here. So this actually spends a lot of time bouncing around on the membrane. So the pH domain maybe not quite so much, but the F0 domain very clearly falls over, and I'll show you some kind of quantitative about what that means later, and exposes this helix, which is interestingly a huge source of protein-protein interactions. So this helix kind of going from a vertical, maybe hidden, to like a horizontal, less hidden interaction. Could very well be interesting, real biological phenomenon that we're actually looking into. So just kind of set up what we simulated here. All of the bilayers were 70% PC, and then either 30% PS with one pip. And then it became very clear after simulating some of them in both situations, where basically 5% of the PS is replaced with pip, that there were enough interactions outside the primary binding site. We did most of our, I'm going to show you the analysis from the 5% data overall here. All right, so basically what we're seeing here are the orientations of the pH domain in relation to the lipid bilayer. And so basically the pH domain, as we saw, spends a lot of time sitting sort of vertically. The contacts you see here in blue are primarily inside the canonical binding site, although you do see a little bit of interactions. And then, like I said, the total number of pips you see actually kind of sits around 2 for the pip2, and then it gets higher for the pip3. The parameter we're adjusting here is the overall net charge of the pip head groups based on the protonation state. Another interesting feature we kind of identified here were that the calciums, which we put in there to sort of accentuate the negative charge of the PS and pips, shows very clear interactions bridging the negatively charged side chains of the pH domain with the negatively charged surface of the membrane. So this is a very soluble protein and has a lot of charges on the surface. And we've been looking for a long time at how the positively charged side chains interacted with the negatively charged lipid surface. What we found out is that the calciums actually allow for similar interactions between the negatively charged PS. So something very similar to what we saw with PS-PS interactions being mediated by calcium. The actual sites of interaction, like I said, are sort of focused along the primary binding site. A lot of Ks here in the list of highest contacts. The thing that kind of jumped out to me looking at this data is that the profile between PIP2 and PIP3 are very, very similar. So this is what I was talking about at the beginning. There's not a huge reason from this data to expect there to be a big activation difference between PIP2 and PIP3. And so actually understand the difference between those two, we had to look at the energetics. And so the PIP3 is a much tighter binding. It's got much lower energy, despite the fact it's interacting with basically the same residues, which was very interesting and gives us a lot of things to think about as we move forward into our NMR experiments. The situation for the F0 domain is different, and it's like I showed you in that short little blurb. It actually spends a lot of time parallel to the membrane. That's fallen over conformation. Conformation's stabilized by these interactions that are occurring outside the canonical binding site. And then again, a lot of contacts outside of the one that sort of started out as the originally bound PIP in the simulations. And so again, a very similar interaction profile between the two. So that the primary difference between the binding interactions are not the residues that are being interacted with so much as necessarily the strength of those interactions. Although you can see in the PIP3 case here some extra interactions which sort of help support these interactions, help support that fallen over situation. And so the idea here is that we can now take these simulated contacts and use them to look at with NMR experiments as the sort of wet lab part of the group catches up with where the simulations are. And so you have this time averaged which residue is interacting with nitrogen. And so the idea here is to try some nitrogen phosphorus sort of readout experiments where that dipolar coupling depends on the distance between those two nuclei, and these are nice, short 3-4 angstrom distances. With those high gyromagnetic ratio of phosphorus, we expect that to be a pretty strong interaction. We've tried a couple of times and do not yet have good PN reador to show you. I think we'll keep working on that. I'm confident we'll get somewhere eventually. But basically, here's an example of the same situation, only with carbons. This is actually a natural abundance carbon in the lipid itself. And you can see that the close one dephases, and then far away one does not. So that's sort of our proof of principle. But as we continue to work on our resolution of our spectra, I'm hopeful that we'll be able to get some good 2D data to show you next time I talk to you about this. Okay, so like I was pointing out, that helix, this alpha-1 helix of the F0 domain, is the primary site for paxillin and actin, which are Kindlin-2 protein-protein binders, to interact with. And so it's a subtle effect we saw here in our simulations, where basically the principal component analysis for all three of the sort of less charged pips showed a fair bit of motion along this helix until you got to the fully deprotonated pip3, so the most negatively charged one, which showed dramatically less motion along this helix. So in addition to the falling over and kind of letting that helix become exposed, it also started moving less, and that was on its own. So one of the things we're kind of looking forward to when we start to be able to do some NMR on this domain is seeing if that effect is recapitulated in the NMR, and then of course seeing how that affects binding to protein-protein interaction partners. All right, so we wanted to think about the dimerization as well. And so the idea here was that we can't do all atom simulations of the full-length Kindlin-2 dimer. It'd be just too many atoms. And so what we've done here is coarse-grained simulations. And so the idea here is you took the crystal structure that was already in existence, you kind of modeled in using modeler the pH domain. And then the idea here is to run these coarse-grained simulations with PIP actually bridged with a sodium. This is the Martini model, so everything's a big circle. Multiple atoms are kind of mixed together with a sort of charge that is responsible for getting those sort of charge interactions. And so the idea here is we're going to pull the monomer and dimer Kindlin-2 off the membrane to try to understand if this dimer is more tightly bound than the single domain on its own, to give you understanding if the PIP2 to PIP3 transition helps us understand, support the bound state a little bit more than would just be seen from a single Kindlin-2 bound to the membrane. Try to understand if this crystal structure, again, and here's that citation I was telling you about, this paper has just completely revolutionized the Kindlin field. Everybody's been trying to understand what this means in the years since it came out. And so this is a pipeline we've kind of are working on for full-length Kindlin-2, which is sort of built off something Mark Sampson and company worked on for just pH domains. So they did a set of experiments to sort of understand how pH domains bound membranes. And so we're trying to sort of generalize it out to a much larger system in this Kindlin-2 here. All right, and I'll skip that slide. I don't think we need to think about that. All right, so one quick thing we've been working on the 100 kilohertz. How much time do we have, John?
If you could wrap it up in about 5 minutes, that'd be great.
Sounds good. My PowerPoint's been open 7.5 hours, so the clock on the top isn't helping me. Basically, what we're doing here is using a long CP period. So one of the things we figured out is that the HC cross-polarization time in a HCONH and a deuterated sample requires long-range interactions. So why don't we use that to measure things? And so what we did, lower spinning with Daniel, who was a PhD student I was supervising in Hartmann Ashkenaz's group back in Berlin, is basically turn that into a 3D experiment that lets you see these cross beta sheet strands, hydrogen bonds, basically, where basically the closest proton to CO in a beta sheet is the one across the sheet. And so you expect to see very strong peaks to your neighboring residue, but also to the one across the sheet. So you can see here, the 25 and the 26 are the strongest peaks, but the next strongest peak is 15, which is the one right across the sheet. And so that's something we recently published, like I said, with Hardmet on the slower spinning data, and are actively working with on the faster spinning data now here in at Rutgers. All right, and so I'm going to quickly go through some of these materials type projects because I have a couple of minutes. And so the first project is about drug release from polymer nanoparticles, and so that's recently been submitted. This is the work of Catherine Miles, who is a PhD student in Adam Gormley's group, who's actually in chemical engineering here. And so the idea here, what she wanted to do was understand how drug release from polymers, like what parameters is it based off of? And so basically she started out on a sort of exhaustive review of all of the different parameters you could measure on these polymers, and then measuring the drug release to figure out what happened. And so how we got involved is basically she wanted everything she could think of, and one of the things she could think of was NMR, so she talked to us. And so basically what we did was took the two polymers that were kind of at the two edges of the drug release. So the one that was completely releasing after 100 or so days, and the one that was only releasing about 40, and actually did solid state NMR on them. And so I've thrown all of the data from that paper into one slide, because the moral of the story is we saw absolutely no changes in the chemical shift of the drug and the bound state. None whatsoever. So here's the fluorine. It's a fluorinated drug compound. Chemical shifts are the same. Here are all the carbons. The difference between the drug-loaded spectra and the polymer spectra is you literally, if you wanted to, you could just add these two together in Mesternova, and you would get this spectrum. There are basically no chemical shifts of the carbons, the phosphorus, or the protons, chemical shifts, deltas, upon interaction with these two objects. And so basically what that led her to think about is what the mechanism for the different in release, because these are very, very dramatically different. And so what they started thinking about is how big the pores inside these polymers are. So they took some PXRD data and actually able to look at the crystallinity of the drugs. What you're seeing here in blue is the actual drug signals in the XRD data. And you see it's much more crystalline over here, much sharper peaks. And so basically the answer to the question was that the pores are much bigger in one of the polymers than the other. And so we did an NMR experiment to help determine that. And so basically the melting point of water inside a constrained environment changes based on how big or small that environment is. And so normally you would just use 50 for this number, but because she had measured every single parameter you could think about for these things, we're actually able to go out and calculate them and get a real number. And so basically what we determined is that at the coldest temperature we took these experiments at, which is 252 Kelvin, 1% of the water in one of the polymers doesn't freeze at all. And so if you plug that into the equation, that means those pores are smaller than 4 nanometers, whereas there's no extra delta at the top end, which means that the bigger pores are at least 30 nanometers in diameter. And so actually that difference in ability to escape is literally just a path length argument. There's no direct interactions between the drug and the polymer. It's literally just a longer maze for the polymers to come out with. And that's that conclusion. And then just to talk about Ashley's MOFs for half a second, we're basically studying a metal organic framework here. We did the assignments using 2D HC correlations again. And what we wanted to understand is how this thing was capturing CS2. And we had an amusing serendipitous discovery that, well, we basically initially studied the CS2 peak as it got captured, but this extra peak grew in. And Tom Pop, like I said, the postdoc who has experience in MOFs, I looked at that and said, that looks like CO2. And so basically they opened the rotor up and they noticed something smelled. And so they used their nose gas spectrophotometers to identify that as perhaps H2S. And so we're actively doing experiments with slightly more sensitive techniques to determine exactly if that is. There's lead acetate paper in the wet lab as we speak. And that seems to be what's happening. So hopefully you will see a paper on that coming out soon, where basically in addition to capturing CS2. This MOT's actually converting CS2 over to CO2. Something that you would want to do, as this is toxic and this is only the greenhouse gas. So with that, I hopefully was able to talk to you a lot about our PIP work, a little bit about our methods, and a little bit about the materials. I need to thank my group, of course. Jackie did the proton detected experiments and all the protein expression. Here she is with the 600 and our helium recovery experiment here that is not yet installed, but we're actually looking quite forward to. Ashley does the lipid on their own, and then of the polymer and MOF experiments. Robert does doing all the simulations. He actually hasn't been on campus since March of 2020. And like I said, Tom, the new postdoc. Jackie, looking forward to doing postdocs in other people's labs. So if you are looking for a postdoc who likes to do impossible things without a lot of leadership, hit her up. And of course, thank you guys for listening to me.
Well, thanks, Andy, for a wonderful talk. Lots of very interesting things going on in your lab. I think we have maybe time for one or two very quick questions, and I'll open up to the floor. If anyone has one, you can just go ahead and unmute yourself. If not, I can ask one real quick. So in your P31 spectra that you showed at the very beginning of the PIP, you had the nanodisc spectrum at the very end that was significantly broadened compared to everything else.
Yep.
Do you think that's just a factor of the restricted motion of the lipids in that nanodisc itself?
So one of the things we're going to work on here moving forward, so these are what we like to call medium-sized nanodiscs. And so there's like 90 lipids on this surface, and so they are restricted. If you remember from the nitrogen 1D spectrum, the nitrogen side chain peaks broadened out as well. So I'm wondering if we're capturing a bunch of slightly different conformations, because this is the really cold temperature. This isn't -40.
Okay.
And so I'm kind of hoping that that is like 10 really sharp peaks all on top of each other, and not that we're broadened all to get up, but we haven't gotten to take the 2D spectra yet to check on that. So I'm kind of looking forward to getting that done, but I'm thinking that's going to have to wait until we have deuterated proteins. That's sort of the next step on this, is to do this whole experiment again with deuterated binding domain. And of course with the F0 as well. So everything I showed you today was on pH, because the F0 is a little further behind. We don't have that protein in hand yet.
All right, thanks. Are there any other questions?
Hi, Andy. It's Bernie.
Hey.
Really great work you did. I have a question about the water and the polymers and the different porosities that you chose. And when you looked at the different types of the water, did you ever think that there was different exchange motifs going on with different maybe conformational populations of different types of water?
I mean, so we definitely have multiple water populations. So this is like the in-pore waters. There's a bulk water signal that goes away at the normal water freezing temperature here at 273. They wanted to understand if this water would ever freeze. And so this actual NMR tube was in liquid nitrogen prior to going into the magnet. The facility manager wasn't thrilled when he found out about that. So we froze that. We took that really cold before putting it into the magnet, and that peak never went away. So as to whether or not it's exchanging, yeah, I don't know if that answers your question.
Do you think that there could be a high order exchange that could give a sharp peak, where you could still have a solid water and still have a high order exchange rate and still have a solid state?
I mean, I have seen bound waters in protein nanocrystals, and they're much broader signal than that.
Tetrahedral or octahedral motion, for instance.
That is not something I have any real familiarity with, so I can't answer your question, I don't think. Something to look into, though. I'll think about it.
Cool. Very cool. Thanks, Andy.
All right, so I think in the interest of time, if there are any other questions, we can take that to the chat, and Andy can keep an eye and answer those. But let's go ahead and get Dave Rodniak set up here for our